Only one cotyledon exists in the case of monocot embryo. That is why it is called as a monocot. Endosperm should get formed first because the endosperm is the nutritive tissue that provides all the nourishment that is needed for the developing embryo. Scutellum is nothing but the entire cotyledon of the monocot is called as the scutellum. So all these green shades that are there, it is the scutellum. Hello everyone, a warm welcome to this session and concept class for class 12th biology. I'm Dr. Divya, biology faculty. So in this session, we'll learn about the post-fertilization structure, that is after fertilization, what are the structures that develop inside the embryo sac session. So first we'll talk about the post-fertilization events. So what are the important events that occur after the fertilization? So fertilization means the fusion of the male gamete with the female gametes, right? Here the male gametes came from the pollen grain. That is the pollen grain which had two male gametes. Those pollen grain will fall on the stigma. If it is compatible, then it will germinate. It will produce the germ tube or the pollen tube. That pollen tube will travel through the style of the stigma and it will reach the opening of the ovary or the embryo sac wherein the micropyle is being situated. So through that micropyle, it will reach the synergids of the embryo sac and it will deliver the male gametes in the synergid. Now, one of the male gamete from the synergid, it will travel and fuse to the egg cell, and one more male gamete will fuse with the polar nuclei or the central cell. Now, because two times fertilization occur, it is called as double fertilization. So, the events which take place in a flowering plant after double fertilization is called as post-fertilization events. And the steps that are involved in the post-fertilization events or what all occurs in the post-fertilization events are: first is development of endosperm from primary endosperm cell. Now, how did this primary endosperm cell get formed? That is, one of the male gamete will go and fuse with the two haploid nucleus which is present in the central cell, and that central cell will get converted into a primary endosperm cell. Next, what happens? Development of embryo from the zygote. Now, here what happens here is the other male gamete will go and fuse with the egg cell. That egg cell will form a zygote after the fusion. That zygote will develop into the embryo. So that is in the second, that is the next event. After that, the development of the seed will take place, and after the development of the seed, the development of the fruit from the ovary will occur. So we'll study these two in detail in this particular session, and how the seed development occurs and how the fruit development occurs we'll study in the next session. So talking about the post-fertilization structure. So the first structure that always gets formed after the fertilization is the endosperm. That is, the endosperm should get formed first because the endosperm is the nutritive tissue that provides all the nourishment that is needed for the developing embryo. So therefore, before the embryo development occurs itself, first the endosperm should get formed. Now, how will the endosperm get formed? The endosperm gets formed from the primary endosperm cell. Again, where did the primary endosperm cell get formed? It is nothing but one of the male gamete that was there in the synergid that went and fused with the central cell having two nucleus, wherein triple fusion occurred. It is triple fusion, right? Because in the central cell, it had how many nucleus? Two nuclei were there. Now here one more cell, male gamete, went and fused here, and that male gamete will also obviously have its own nuclei. Now triple fusion is occurring wherein it will get converted into a primary endosperm cell having a primary endosperm nucleus, right? So that primary endosperm nucleus, that after the fusion, what it will happen? It will undergo repeated division and a triploid endosperm tissue will get formed. And as I told you, the main purpose of the endosperm is to provide nutrition to the developing embryo. And the endosperm can be of free nuclear or cellular type. So free nuclear endosperm is the one that is most commonly seen in almost all the seeds or during the fertilization, post-fertilization event. So next we'll talk, that was about the endosperm. So from where the endosperm developed, we got to know. Now we'll talk about the embryo. Now embryo, one more male gamete is there which is being released to the synergid that will go and fuse with the egg cell. Once it goes and fuses with the egg cell, that egg cell is now a zygote. That zygote will undergo repeated division to form what? The embryo, and this embryo development and all that will occur at the micropylar end itself because the egg cell is present towards the micropylar end. So the embryo develops from the zygote. Now we have understood that. Now the development of the zygote to form the embryo starts only after the endosperm formation. That I have told you. Why? Because endosperm is the one that gives the nourishment. So before the embryo develops itself, the nourishing tissue should already be ready. So that is why the endosperm gets formed first and then the embryo development starts. Next, we'll move on to the stages of embryo development. So the stages of embryo development in the dicotyledons and the monocotyledons is one and the same. Only the structure tends to vary. That's it. So talking about the development starts with the fertilized egg. Okay, so what happened? Fertilized egg means along with the synergids in the micropylar cell, there was one egg cell, no? There was one egg cell. So say for example, if this is the egg cell. So to this egg cell, what happens? One of the male gamete will go and fuse. That male gamete will become what? It will develop into a zygote. Now this is the zygote. Zygote is a single cell. That zygote will undergo division. It will form a two-celled stage. That two-cell stage again undergoes division to form 8-cell stage. Then it will undergo again division to form globular stage. This looks like a globe. See this portion, it looks like a globe. So that is why it is called globular. Next it will undergo again repeated division to form a heart shape. After the heart shape, again it will undergo repeated division to form the torpedo shape. Then after the torpedo shape, the matured embryo will get formed. So these are the stages of development of the embryo. Moving further, these stages, as I told you, it is the same in both the monocots and the dicots. Now we'll talk about the structure of a dicot and monocot embryo. We need to know the structure because structurally they are different. The dicot and the monocot embryo are different structurally. So the dicotyledonous embryo has the following important parts. The first is the embryonal axis. So the main axis of the embryo, which divides the embryo into different regions, is called as the embryonal axis. So in that, what all lies in the embryonal axis, we will look into. They have the cotyledons. So cotyledons, these are the two cotyledons of the embryo. They have the cotyledons. Next, these cotyledons actually they help in, or the embryo of the dicotyledonous seeds, they help in the storage of food, and they also, the cotyledons in general, in particular the cotyledons, they help in the storage of food and they provide nourishment to the developing radicle and the plumule. So this is the plumule region and this is the radicle region. And this is the hypocotyle. This we can call as the epicotyle. Moving further, we'll talk about the epicotyle and the hypocotyle. So I told you this is the, again I'll write here, epicotyle. Next here, hypocotyle. Then we have the plumule. Then comes the radicle. So epicotyle, it is the part of the embryonal axis which is present above the cotyledon. So if you can see here, these are the two cotyledons. So epicotyle is, epi means top, hypo means below. So epicotyles are present above the cotyledons, and above the epicotyle is present the plumule, and the plumule gives rise to the shoot. And next, hypocotyle region is present below the cotyledons, and the hypocotyle, below the hypocotyle is present the radicle, and the radicle gives rise to the root development. So now we know about the epicotyle and the hypocotyle. Moving further, let's study about the structure of a monocotyledonous embryo. Monocotyledon is embryo. It also has the embryonal axis. It is the main axis of the embryo which divides the embryo into different regions or parts. So, what are those parts? Let's look into. So, talking about these parts, first we'll talk about the cotyledon. So, only one cotyledon exists in the case of monocot embryo. That is why it is called as a monocot. Mono means one, cot is nothing but cotyledon, monocotyledon. And this also has the scutellum. So, this is nothing but the scutellum. So, now we know about the scutellum. Scutellum is nothing but the entire cotyledon of the monocot is called as the scutellum. So, all these green shades that are there, it is the scutellum. Now, in the scutellum, we can find this portion. It is called as the coleoptile. And what is the function of the coleoptile? Same like how we learned in Dicot. It protects the plumule. Plumule is the shoot tip of the monocot seed through which the leaves or the shoots develop. And that plumule, it is protected by the coleoptile. Next, we have the coleorhiza. And the coleorhiza, it protects the root cap, as well as it protects the radicle. So, that is the function. And root cap, it acts as a protective covering. It is thimble-like or it is cup-like, which acts like a protective covering which protects the root tip. So, the root tip contains a lot of continuously dividing meristematic cells which is filled with young cells. So, therefore, they are very fragile. So, they need to be protected as and when the root grows in length towards the soil. So, that protective function is done by the root cap. And in turn, the coleorhiza covers the radicle and protects the. So, this is about the structure of the monocot seed or monocot embryo. Monocot embryo and dicot embryo, the stages of development is same, but structurally they tend to vary a bit. So, this was about this session. So, in the next session, we'll talk about the post-fertilization structure again, that is seed, how the seed gets formed, and we'll talk about apomixis, which is one of the important terms, and then we'll talk about dormancy and we'll talk about the formation of the fruits. So, this was about the session. I hope you found the session useful. So, we shall meet again in the coming session discussing this new topic. So, see you in the next session. Thank you.